pizza.
But not just any pizza, this pizza is going to undergo meiosis.
So what parts of the cell are we going to need?
Well, this is our cell membrane and our cytoplasm.
These look like chromosomes.
Exactly, along with our spindle fibers and centrioles.
Okay, let's get cooking.
So, we begin interphase. The chromosomes and DNA are replicated in the S stage, and each form a pair of sister chromatids. The diploid cell will eventually form 4 haploid cells.
Meiosis is made up of 2 cell divisions, meiosis I and meiosis II. The first step in meiosis I is prophase I.
The cell's chromatid condenses and homologous chromosomes pair up and overlap during synapsis. One chromosome is maternal, while the other is paternal.
The homologous pairs are now visible as tetrads, and crossing over occurs so that each chromosome has DNA from both parents. This is the reason for genetic variation.
As the nuclear membrane dissolves, the centrioles start to migrate to the opposite poles of the cell and spindle fibers form.
Prophase I is the longest phase of meiosis, and now we can enter into metaphase I.
The homologous chromosome pairs arrange themselves along the equatorial plate in random orientations.
Spindle fibers attach to each chromosome and prepare to pull them to opposite poles of the cell.
Then, we move into anaphase I. The tetrads separate and the chromosomes are pulled apart, but the sister chromatids remain attached at their centromeres.
Now it's time for telophase I. The chromosomes have now migrated to opposite ends of the cell. A new nuclear membrane begins to form around them.
The cell begins pinching in and forming a cleavage furrow, and eventually splits into 2 haploid daughter cells. This process is called cytokinesis.
Now that meiosis I is done, we can move into meiosis II. We start with prophase II. The centriole in each daughter cell duplicates and forms new spindle fibers.
The chromosomes condense and the nuclear membrane dissolves. We're finally ready for metaphase II. The chromosomes line up along the equatorial plate and spindle fibers attached to each sister chromatid.
Now it's time for anaphase II. The chromosomes split apart at their centromeres as the spindle fibers begin pulling the sister chromatids to the opposite poles.
If the sister chromatids fail to separate, this is known as non-disjunction. This can result in one of the gametes having an incorrect number of chromosomes. Unfortunately, this can cause genetic disorders such as Down syndrome.
Time for telophase 2. Now that the chromosomes are at opposite poles, a new nuclear membrane forms around them.
Cytokinesis occurs again, and we are now left with 4 haploid daughter cells.
Well, that was fun, but I think it's time to get these cells in the oven.
Yeah, they look delicious.
See you next time on K&A's Kitchen.